What is up, guys? So it is currently 5 in the morning, and I have an exam at 9:30. So that's about 4 hours from now. So I woke up early because today is actually my last regular anatomy exam before our final, and it contains a lot of information. So it's going to be on the special senses of the human body and the endocrine hormones. So what I'm going to be doing today is I'm going to be going over all my notes from leading up to this exam, all the pages leading up to it. A bit of the textbook right here, some figures. I'm going to be going over this practice study guide. I'm going to be making a video of an online study guide I made to go over. I'm also going to be going over all the endocrine hormones that I had to memorize for this exam to prepare. And I'm also going to be using this whiteboard to write down some of the topics are hard to understand, and I also have a practice exam. Okay. Okay, let's jump right into it. So the first figure we're going to be looking at today is figure 15-1. You guys already saw this in the previous video when we're studying for the quiz. So this is an overview of events occurring along the sensory and motor pathways. So we have a stimulus come in, and there's a depolarization of a receptor. Produces a graded change in the membrane potential. Then an action potential is generated once depolarization happens and reaches a threshold. Action potential develops in the initial segment, and then propagation happens. It acts on the sensory neuron, carrying information about the type of stimulus as action potentials to the central nervous system. So the type of stimulus can either be touch, pressure, or temperature. So stimulus comes in, depolarization, action potential is generated, and then there's propagation by sending that stimulus to the central nervous system as an action potential. Then central nervous system is processing. Information processing occurs at every relay synapse. Information will be distributed to multiple nuclei and centers in the spinal cord and brain. So if it's a voluntary motor pathway, if only about 1%, the arriving sensations are related to primary sensory cortex, and it can enhance an involuntary response. So an involuntary motor pathway response is more like a reflex, reaches the spinal cord and brainstem for a quick response before the sensations reach the cerebral cortex. Okay, the beginning of the circuit. At some point, a stimulus arises at a receptor. I'm reading the notes now. The organ receiving the stimulus is the receptors. Sensory receptors of various types. So receptors are divided into 3 areas: special sensory, visceral receptor, and somatic skin receptor. Somatic comes from muscles, joints, and skin. Stimulus arrives at a receptor. It must reach a threshold, the stimulus through a chemical gate. An action potential, which is propagated at the central nervous system where there is processing. If it is a sensation that makes it all the way to the cerebral cortex, it becomes a perception. A sensation that makes the cerebral cortex becomes a perception. A sensation is a stimulus that is received and makes it to the central nervous system. Its effect may go on similar to a reflex. To be aware of it, it must reach the cerebral cortex. Conscious and subconscious parts of science, perception versus sensation. Afferent and efferent division is based on the intensity of a stimulus coming in as loud or a weak sound. Intensity comes in as a stimulus. It can be strong or weak. It's based on how many neurons are receiving information, or it can be a rapid firing neuron reaching threshold very often, or a lot of neurons honed to the same receptor. Afferent division, intensity of stimulus comes in as loud or weak sound. An intense stimulus versus a weak stimulus is based on how rapidly neurons are being fired down. Stimulus intensity is transferred through a frequent impulse on some neurons. General senses, as opposed to special senses, are senses laid out throughout your body that somatic has a field sensitive area. One spindle fiber is muscle relaying information about just a certain group of muscles. So we have to read about the general senses over break. This was before spring break. There's some old information. Wow. March. Okay, April stuff. Same figure, so sensory input / the cost part of the story. Involuntary and voluntary response if it is not visceral. Visceral is more on the organs. So there's 3 types of senses: general, somatic, visceral, and special senses that bring out an action potential that gets sent to the central nervous system for a reflexive or voluntary response. How do you classify senses? As general, having somatic origin. General meaning somatic, versus special sense organs. General senses cover the senses of touch, pressure, pain, and proprioception. Pain, temperature, vibration, and proprioception. Proprioception is the position of muscle extensions of your joints. Sensory organs include Golgi apparatus. Joints have sensors that sense how much they have been moved. General senses include touch, pressure, pain, and vibration. Received by receptors. Receptor types include mechanoreceptors, which mechanoreceptors consist of touch receptors, pressure, and baroreceptors. Okay, so this is the figure of the receptors and the receptive field. So there's receptor field 1, receptor field 2. Each receptor cell monitors a specific area known as a receptive field. That's where it's receiving information. Chemoreceptors sense temperature. So they're pretty general ones all over the skin below the epidermis. In 2 different places in our body, such as the skin and the hypothalamus. They are sensed by the hypothalamus. Thermoreceptors in the hypothalamus ensuring proper temperature. The other receptors include proprioceptors, which okay, you can't forget it. Thermoreceptors are sensed by the hypothalamus. Skin, hypothalamus. Proprioceptors give a sense of body position. Nociceptors give a sense of pain. Photoreceptors give or from the retina of the eye, receptors on the retina of the eye. Chemoreceptors are a sense of smell and taste. Mechano is more on equilibrium of inner ear and hearing. So there's mechanoreceptors, thermoreceptors, photoreceptors, chemoreceptors, nociceptors, proprioceptors. Noci meaning pain, photo meaning vision, chemical meaning smell and taste. Okay, next figure. This figure on the tactile receptors in the skin. So there's free nerve endings, root hair plexus, hair, tactile disc, different corpuscles, receptors. I'm really trying to do good on this last exam because if I do good, I get to have a smaller final exam at the end of the semester. Okay. So we don't need to know all the different receptors on your skin, only need to know these. Baroreceptors are in the brain and the digestive tract. Baroreceptor would be a type of mechanoreceptor. Chemoreceptor is in or near respiratory centers in the medulla, sensing oxygen, carbon dioxide, regulating pH. Baroreceptors in the lungs tell your pons pneumataxis centers to know how to expand your how expanded your lungs are. Okay. And we moved on to chapter 17 already. So first figure in chapter 17 is this olfactory organ. As I said before on the previous videos, we have olfactory epithelium right there, the nerve fibers, the bulb, the tract, the central nervous system right there, and a bit of the epithelium right here. There's the bulb, there's the olfactory gland, the basal cells, some olfactory dendrites. Yep. So conic versus phasic receptors. When information comes in at a greater strength, the interval of impulse is sped up. When it comes at a lesser strength, the interval is slowed down, but they are sensing constantly. Conic receptors receive information at a regular interval. Phasic receptors sense changes at a periodic interval. Adaptation are different senses in your body. It's when different senses in your body become accustomed to a stimulus at different rates of speed. Example would be fast adapting senses, not aware of any more incoming sensations, such as touch, pressure, and smell. Slow adapting senses means you're still aware of the sensations for a long period of time. An example would be pain, body position, fatiguing away from this. Okay, next figure is on olfaction and gustation. So smell and taste. Olfactory receptors are dendrites, specialized neurons. When they come in contact with those chemicals, there is a generated potential or depolarization. It gives information about our environment, although through the sensory information is diverse and complex. Each special sense originates at a receptor cell that may be neurons or specialized receptor cells that communicate with sensory neurons. So you can see here, once there's a stimulus, reaches threshold, there's a generated potential, action potentials right there. These are all considered action potentials once you pass the threshold. So this other stimulus is received. It's received at dendrites, goes to olfactory neurons, then down axons to the central nervous system. For gustation, receptors of taste, vision, equilibrium. These use synapses, or the process of sensory neurons. Triggering the release of chemical transmitters at the synapse. These transmitters then depolarize sensory neuron, creating a generated potential and action potential. So there's a receptor cell that gets stimulus, depolarization, and then another stimulus at the axon. So stimulus to receptor cell, to the synapse at the dendrite, then at the sensory neuron, then at the axon, there's a generated potential and action potential is formed. Oh dang. A stimulus is picked up by a neuron, receptor cell. Stimulus hits a specially sensitive cell, which then speaks to its neuron. We don't need to know G protein, cyclic AMP. When a stimulus arrives at a receptor cell, the input grades up to a threshold. Receptor potential goes up to a graded potential. Graded potential summates to make an action potential. At the initial stimulus level, the graded potential is called a receptor potential. Receptor potential goes on to make an action potential. Back on the figure of the olfactory senses, the olfactory sense is the least evolved sense. Its ability to sense chemicals in the liquid environment. As a molecule of odorant enters. As a molecule of odorant enters your nose, the odorant is picked up by olfactory dendrites. Odorants are chemical molecules that are in a sense of smell, that need a level of moisture in order to smell the odorant. For all the special senses except vision, know the anatomy of the collecting structures only to the level of the cells. So types of receptor cells and their structures. Airborne molecules get dissolved and form an action potential. Olfaction has one component that goes straight to the hypothalamus. Olfaction is the one special sense that is direct. Olfaction is one component that goes straight to the hypothalamus. Collection is the one special sense that is direct. Olfaction. Olfaction has one sense going straight to the hypothalamus. Olfactory nerves lead to the olfactory tract going straight to the hypothalamus. Olfaction can go straight to your memory and emotions. Direct special sense neurons become nerves that go into olfactory tract, leading to the hypothalamus, triggering memories and emotions. Conscious awareness doesn't become a perception. It doesn't become a perception, it makes it to the central nervous system. You don't become aware of it until it makes it to the cerebral cortex. The trip takes longer than a trip to the hypothalamus, associated with other memories. Smells are not like taste, smell, discriminating one odor from another. Next figure. Next figure is on gustatory receptors, so taste. So we have taste buds on our papilla. Taste buds, papilla, locations of the papilla, foliate papilla. Yeah. Taste papilla are little bumps you thought were your taste buds on the surface of your tongue, in the back of the pharynx. Dissolved in saliva, while gustatory taste buds process and form an action potential. Microvilli are taste hairs that take the taste in, and it becomes a neural message that the axon takes and forms cranial nerves. There are different shapes of papilla, spongiform like fawns, filiform like a leaf. Papilla have a crack in each side where saliva is kept and dissolved in saliva, and the taste and molecules, they sit in cracks until taste buds take them in across the membrane for the formation of an action potential. The inner ear is responsible for equilibrium of rotational position and acceleration of up, down, forward balance. Has three semicircular canals. Middle ear is involved in detection of sound in the cochlea, mechanical bending, moving, turning. Other types of equilibrium takes place in the vestibule. When info from three different semicircular canals give position of the body in space. Okay. Three special sense structures include the cochlea, the vestibule, the central canal for rotation and body position. Hearing relies on all three of these. Semicircular canals are filled with ducts. The fluids inside the ducts are called lymph. Canals are bony structures and ducts are socket tubes. Canals are Canals are filled with ducts. The fluid inside the ducts are called lymph. Canals are bony structures and ducts are socket tubes with lymph fluid. Perilymph, endolymph, not just water, lymph fluid. Also water allowing them to work right. Most of these lymph, including, are completely thickened, not just like water. The essentially movement of fluids at 3 canals triggers ampullae. Ampullae are disturbed ends of each canal that contains inside each jump structure a thickened form of lymph fluid. I'm fatigued. It's starting to hurt. Ampullae has a thickened form of lymph fluid. The gel endolymph fluid gets thickened into a gel. The gel bends the hair cells that are bent to a certain degree, as the cupula will pull on them, and that tells your position in space. This tells you your position in space. Tilt, bending gel, and bending hairs. And then we had a quiz on that type of material. It was on so what were the 5 main types of receptors? Nociceptors, thermoreceptors, chemoreceptors, mechanoreceptors, photoreceptors, and proprioceptors was an extra credit for that one. Transduction is a translating, duction equals signaling. Modalities are stimulus, sound waves, vision, light waves. The body needs to translate these stimulus into a specific language of electrochemical conducting from a neuron as an action potential. So there's 2 special chemical senses: smell, olfaction, and taste, gustation. So back on the same figure of the tongue that we were looking at. Humans have a basic taste ability, so 5 senses: salty, sweet, bitter, sour. And umami. Umami means savory, identified scientifically as a taste of protein. As a savory as opposed to sweet. Some adaptive significance: taste changes by gender, age, and physiological state. Why do we have taste? It has to do with survival value, evidence to adaptive significance. During reproductive years, we're sensitive to taste. Taste in infants and newborns changes a lot in the first few years. Infants have heightened ability to taste bitter. Infants have an amazing olfactory ability. Innate ability for sweet taste. Sweet means glucose. Salt craving used to be associated with a large body of materials that are critical for our body processing. In human evolution, salt is still collected when come across in the desert. Okay. Next figure is on equilibrium. Okay, here. So the anatomy of the ear. Okay, so external ear, the anatomy of the ear, external ear, middle ear, and internal ear. So there's the external ear with an auricle, some cartilages, an external acoustic meatus. There's a tympanic cavity with a tympanic membrane. It's also called your eardrum. Auditory ossicles and an oval window. And that oval window is connected to the vestibule. The vestibule is connected to semicircular canals, which have lymph inside them, thickened fluid. And if we go even closer to the internal ear, we have the snail, or the cochlea, and we see the facial nerve and the vestibulocochlear nerve there. So round window is not too important, oval window is the more important one. So equilibrium. Equilibrium takes place in one small portion, the vestibule, and 3 semicircular canals. Comes in 2 types, in 3 semicircular canals detect tilt and position of the head. The inner ear begins where startup meets the oval window, where the stirrup meets the oval window. When the oval window vibrates, innervated by the stirrup, the liquid gets vibrated in the vestibule, and the duct containing the lymph inside the soft duct is endolymph. It takes on a slightly gelatinous texture once in semicircular canals, once it goes down into the vestibule, because where you have hair cells, the hairs are being moved and bent, and mechanoreceptors sense these. Sensitive hair cells are sensitive long hairs containing cilia bent by a thick endolymph so that it doesn't slosh. It just moves in a certain direction. Next figure. Next figure is of the internal ear. You can see in this cross section of a semicircular canal, perilymph, endolymph, right there is the gelatinous stuff. Here we can see the semicircular ducts, the anterior lateral post. There's the vestibule. More specifically on the vestibule, you have your utricle and your sacculae. The ducts, tympanic ducts, the cochlea, the snail. Cristae with ampullae, the maculae, and the lymphatic sac. Sensory receptors include the cristae, maculae, and spinal organs. Okay. The inner ear has hard ducts with a soft tube. Endolymph, it takes a thickened liquid form in the vestibule. It is a thickened form version of water so that hair is not just flapping. Endolymph takes a thickened liquid form in the vestibule. It's a thickened version of water so that the hair is not just flapping. So there's sensitive hair cells that act as receptive cells that help your brain be consciously aware of your position in space. Hairs are thickened by an endolymph that moves as your head moves. Okay. Okay, there's going to be a lot of figures now. Okay, 17-24C. You can see here the endolymph movement along the length of the duct moves the cupula and stimulates the hair cells. So we can see there's the semicircular duct, there's the cupula, and there's hair cells inside it. This is when it's at rest. This is when it moves here. The direction of relative endolymph movement is going there. Moves here, endolymph is going there. Ampullae or crista ampullaris. The utricle is close to you, which looks like a semicircular canal. The sacculae is close to the snail. Okay, next is this one. This figure right here shows a representative hair cell receptor from the vestibular complex, bending the stereocilia toward the kinocilum, depolarizing the cell and stimulating the sensory neurons. Displacement in the opposite direction inhibits sensory neurons. So a hair cell, stereocilia up there, displacement, sensory nerve endings connected to the hair cell and supporting cell. So mechanisms of utricle and sacculae have sensitive hair cells linked to neurons, yellow, which join to axons to form a nerve. Vestibulocochlear nerve would be an example. Okay, next figure is of the sacculae and utricle. So you can see there's a sacculae, there's a utricle. Utricle is near semicircular canal, sacculae is near the snail. The ducts. And these are the otoliths, which are on top of the hair membrane, the nerve fibers. So structures of individual macula. So if our head is in the normal upright position, the otolith would be above the hair cells, the stereocilia. If it's tilted posteriorly, otolith moves downhill, distorting hair cell processes. Receptor output increases. Hair cells join together with axons forming a vestibular cochlear nerve. Otoliths is a thickened molecule. They're like little stones. Lymph is a stone. Otoliths, and you move the little stones that move the stones and gels to trigger a movement in the receptor cell, consisting. An example would be going down or going up stones. Float. Back to that figure of the hair cell. Stereocilia in lab panels. Okay, 26. Next figure is on pathways for equilibrium sensation. So we can see semicircular canals, vestibule. So as those hair cells are moved, signal is sent down Vestibular ganglion are nerve fibers to the cerebellum, vestibular nucleus, and sent to the pons or vestibular spinal tracts, and different nerves in the body. Vestibule comes to pons and up to the cerebral cortex. To the pons and up to the cerebral cortex, where information is used by the cerebellum, to adjust cerebral output. The vestibule joins with the cochlear branch, forming the vestibular cochlear nerve number 8. The inner ear is sterile and has microbes that don't belong there. Middle ear. So the middle ear, the figures of the middle ear, we can see the auditory ossicles, malleus, incus, and stapes. Stapes hits the oval window, tympani, stapes. Different pictures of them. Yep. Okay. The middle ear is prone to lots of infection because the middle ear, although protected by a tympanic membrane, sits between the middle ear and the external ear. The development of these bones is in a more horizontal position. It's not meant to be fluid-filled, but air-filled. Eustachian tube, so there's 3 bones, malleus, incus, and the stapes. Transfer vibrations or sound waves. Transfer vibration sound waves transfer and amplify vibrations from one side to the oval window, where vibrations are sent to the inner ear. Molecules of air are pulsed there. Page 29. Next figure is of the amplitude and wavelengths of sound. Not very important. Sound waves are transmitted to the inner ear as they head through the external inner canal as they set up windows in the middle ear. The inner ear is ready to take the form of waves in a liquid. Wavelength is the distance between 2 peaks. Peak to peak, it's related to the frequency of the wave. Frequency is how many times you reach a peak during a period of time. Amplitude is the height of the wavelength, the depth of the wave. If it's a long wavelength, it means it's a short frequency. If it's a high wavelength, it means it's a high frequency. Fatiguing. We're making progress. So in the microscopes in the lab, we saw the cochlea. Cochlea is vestibular membrane, basilar membrane, and tectorial membrane. Round window is down there. To the round window, these are the stapes at the oval window. Cochlear duct, cochlear branch, vestibular branch. Cochlear branch leads from the cochlea, vestibular branch leads from the vestibule, forming vestibular cochlear nerve. Basilar membrane, temporal bone. So the cochlea has basilar and tectorial membrane, and it has the oval window and round window. You can see the vestibular cochlear nerve there. Okay. Cross section of the cochlea between spaces is a basilar membrane that is a mechanism of transduction. Somatic hair cells, sound waves come into the liquid. The organ of Corti within each little gray patch is a basilar membrane dividing the two spaces. Double hole spaces, or the two spaces per loop of the snail. The membrane that runs between the two spaces are called basilar membrane. Mechanisms in the inner ear are called the organ of Corti. Okay. Next we see the spiral organ, the cochlea again, the basilar membrane, tectorial membrane up there, cochlear wall, spiral ganglion, two-dimensional section of the cochlea, cochlear branches. So we have a tectorial membrane right there, basilar membrane right there, some outer hair cells below the tectorial membrane, and some inner hair cells, and some nerve fibers connecting to those hair cells. Oh man. Hair cells, sound waves come into a liquid and bends a membrane, working its way around the round window, pushing against connective tissue cells, bending basilar cells. Distinctions help brain distinguish between different types of sound and their pitch. Wave now in liquid, pushing on the basilar membrane, pushing on hair cells connected to the bending. Every time it hits the basilar membrane, causing hair cells to bend at a certain degree. So we're going to be talking about frequency discrimination through stapes movement, round window movement, and the basilar membrane being distorted The basilar membrane is in the cochlea also. Distortion of the basilar membrane bends hair cells telling the brain these neurons that it is a loud sound. Amplitude of the wave bends the cells more. The auditory cortex reads some as a soft sound that is not enough to reach the threshold that some humans cannot hear. Loud sounds cause the basilar membrane to bend a lot, causing hair cells to bend more. Amplitude registers to the brain as loudness or volume. Amplitude register brain as loudness or volume. The auditory cortex reads some as a soft sound, but that is not enough to reach the threshold that some humans cannot hear. The pitch story. So the pathways to the for auditory sensation, we have a cochlea going down the vestibulocochlear branch, vestibulocochlear nerve, to the pons. To the pons. Some of the information goes to cerebellum. The rest goes to inferior colliculi, giving motor responses. Ascending acoustic information going to the geniculate nucleus of the thalamus. Frequency sounds and high frequency sounds are delivered to the auditory cortex. To the reticular formation, mononuclear, the cranial nerves. The pitch story is based on the frequency of the wave, based on how many ups there are in a unit of time. High frequency, short frequency waves hit two membranes so rapidly that being bouncing and dissipates energy early. And dissipates energy early in the cochlea. If the brain gets a high frequency, it is transferred as a high pitch. High frequency sound means high pitch. Low frequency works its way up the stimuli, not dissipating. Here in hair cells, translate as a low sound / low pitch. Triggers bend in hair cell, translate as low frequency sound, translate as low pitch. Heads into the brain, the pons, out the thalamus, and straight to the cortex, to areas associated with hearing. Starts out as a primary sensory gyrus, don't synapse until it reaches occipital lobe. Optic nerve goes up crank and back out. Then the thalamus, third order neuron. Pointing primary cortex, sensory cortex, to associated to sensory organs. Oh dang, fatiguing. Figure 17-4. We finished that stuff, balance stuff, and now we're going into vision, finally. External features and accessory structures. The eye, we can see the lacrimal gland, conjunctiva, lower canthus, orbital fat, inferior muscle, superior rectus muscle, eyelashes, pupil, palpebra, medial canthus, lower canthus, sclera, stuff like that. Okay. Vision, the anatomy of the eye. So the pupil is the region with no iris. It is a hole heading straight into your eye, fortunately covered by a glassy piece called a cornea. The cornea is a transparent portion of the sclera, this bigger eyeball. If all the sclera around the eye becomes white. Accessory structures include the eyelids, lacrimal glands, lateral and superior to the eye. Ducted accessory gland ducts fluid into surface of cornea from lateral or superior to medial of the eye, comes from tear glands to clean and lubricate bacterial side and nourish the cornea. The purpose of tears. Tears contain lysozymes, an enzyme that breaks apart bacterial walls. There's lots of nutrients as well. Parasympathetic division of the nervous system gets involved in the eye by stimulating tear glands. Based on resistance, tears are based on resistance because irritation or sad. Next figure is of the pupillary muscles. So we have a pupillary constrictor muscles around the sphincter. We have circular radiating muscles. Pupillary dilator muscles are more radially from the edge of the pupil. Contraction of these muscles enlarges the pupil. Decrease light intensity. For dilation, you need sympathetic stimulation. For constriction of the pupil, you use concentric circles, concentric muscles. When these sphincter muscles contract, the diameter of the pupil decreases, increase light sensitivity, light intensity, and increase parasympathetic stimulation. Okay, how else is parasympathetic division does what with the eye? Constricting, not dilating. Muscles in the iris are laid out in two directions: circular inner, radial outer. Circular layer is one that is contracted by the parasympathetic nervous system. Sympathetic nervous system dilates the pupil when contracted. Sympathetic nervous system radiates. Smooth muscles dilating pupil for a contraction allows light in at a greater or lesser degree on the parasympathetic innervation, which is contracted circular muscles. Next is this figure of a section of the anatomy of the eye. You can see the fornix conjunctiva, an eyelash, pora serratia, the cornea, the pupil, the lens, the iris, limbus, the fovea, the optic nerve, the retina, the choroid, and the sclera. Here you can see the lids, lacrimal gland, iris, pupil, cornea. The iris is in the back, regenerates color. The iris is backed by a narrow pigmented layer. The function is to prevent stray rays of light, photons, from going where you don't want them to go. The eye is designed to project protons away. The eye is designed to project protons away. Should not be bouncing around or you will get lost. Lots of flashes of images. Input, which is not image focused. Ensures light is coming through the pupil. Its function is to ensure light is coming through the pupil to a specific part of the eye. Conjunctiva is a gasket between accessory structures of the skin and eyeball itself. Okay. Quiz results. Thermoreceptor, nose-receptor, chemoreceptor. Oh, dang. I'm almost caught up. Just take a quick break. Do some stretching. Look at exam questions. Okay, so probably look over these practice exam questions for a bit during my break and do some stretching. I'll just do another half to this study session. See you guys in the next one.